Hello, everyone. Today I'm going to talk about some of the chemical modification of messenger RNA, which recently we are doing, and the paper is still under review. Before I talk, as Thomas mentioned, that we really need to educate the next generation, chemists and the biologists, for nucleic acid, for therapeutic in general. So I wanted to show how can chemistry with biology interact with each other, then we can produce the next generation therapeutics. For this, I wanted to show that in order, unfortunately, the pointer can work here, but I can do the laser pointer. Right. So what can chemists do to help biologists to introduce the next generation therapeutics? So as a chemist, and I'm nucleic acid chemist, and as a chemist, we always think, how can we look in the central dogma, which most of you are familiar with, which starts from DNA, RNA, and end with protein. Of course, this been produced in any organism, in living organism, all the time, million of times. If everything going right, which is most of the cases, whenever something gone wrong, then start to have problems with diseases stuff. So how can we know what is the problem and how can we start to think about solving it? Usually the problem, we have to identify what is the problem by biologists with the help of chemists. Once we know where is the problem in this process, we start to design the drug for it to solve the problem. Chemistry can solve most of these problems. So you start to understand the biology, and then you start maybe to diagnose what is the problem exactly, and then design the therapeutic for it. Okay, so for more than 1 decade now, we're trying to help in understanding some of the biology, but also making some of the material. And what we realize that combination between chemistry, which you can do solid phase oligosynthesis, and biology, which you can ligate to do longer, because of course, when you do solid phase chemistry in the synthesizer, there is limitation of the length of what you can produce. If you want to introduce certain modification at certain loci, then you have limitation of making the DNA or the RNA. In biology, we use ligases to conjugate these together. However, ligases always come with some problems, which is either you can't scale it up because it needs large volume, otherwise it will give catenation, and also the problem of having, sometimes it is not compatible with all salt or all modification. We start to introduce click chemistry, which Thomas mentioned earlier. And with click chemistry, you can do the ligation because it is clean reaction and it can be done in water. And we started to do this ligation using this long linker. And this long linker, we started it for nanotechnology application, but also to start to cyclize oligonucleotide to make short kind of catenane as double-stranded. And you can also cyclize to make something called decoy. And we had a lot of application for this in binding to certain proteins for certain diseases. Some publications are here if you are interested in that. We started to move on after that to see how can we use click chemistry, in fact, to maybe for not only to have biological construct, which you can go as long as it is far from the pocket of the protein or the enzyme, how can we introduce replacement for phosphate so it can be compatible with ligases or with also polymerases? So we introduced this linker, which is shorter than the one I showed before. And with this linker, we kind of found the design in the end after a couple of trials. And this design was able to be compatible in bacteria and in human cells to be read by DNA polymerases and RNA polymerases. We managed to make the whole gene, modified gene, with some epigenetic modification and also with some fluorescence. So we managed to make the whole gene in one pot. So you mix your 10 modified oligo, one with alkyne and one with azide, and they all self-templated. And then you produce the gene, which was function in bacteria, and we tried part of it in human cells. So it was function. And your whole gene here has got 8 triazole, but they have distance, large distance, because the whole gene is like 500 mer. So you have this, in each strand, you have like 4 triazoles, and they all were compatible. We applied this also to make library of single guide RNA. So in one tube, you can have a lot of this modification, because for single guide RNA, in fact, most of the bases are the same. So the only part which you change is this part, which you can have as a complement for your target. So we thought if we can have, say you want to have like 100 CRISPR, instead of making all this, and for therapeutic, of course, in future, we need it to be modified because we need it to be stable. So you can make this in very large scale and put all your modification. And then if you have this biocompatible backbone here, you just design the 20 for targeting your gene. And this 20 will be, of course, very pure, very clean, because it is easy to purify. Then in one pot, you can make your library, and maybe you don't need to purify it. We showed that we can just use kits for purification, and it was really stable and more efficient in cells. You can use also the ligation, here's the scoramide ligation, for detection of messenger RNA. So as a nucleic acid chemist, you can do a lot in this field using ligation. As long as you find the compatible one, as long as you find the right polymerase and the right linkage short enough to be read through by these polymerases. Here as well, one review if you are interested in this field. Few years ago, we started to think about these linkages, not from biocompatibility, but from kind of therapeutic point of view. One thing about these backbones, they are neutral. Because they are neutral, we thought, how can we introduce them to, say, ASOs to decrease the number of charge, but also to decrease the number of phosphorothioate. And the phosphorothioate is being known as very toxic if it is in large quantities, especially in ASO. So to replace some of these, so now we are going from the very long, which was our application before, to make very long gene. Now we are down to short, but you will have multi-addition of this neutral backbone. So we started with Isabel Baker, who was postdoc in Tom Brown group before I started my lab in Southampton. So she made this imide backbone, and we tested it, and we found that if we put 2 or 4 of this linkage in short therapeutic oligonucleotide, it improved the delivery without any moiety. So you don't need any capsulation, you don't need any GalNAc or anything. It goes just genetic without any transfection. And this was like more efficient, like 10 times more efficient than the phosphorothioate and the 2'-homicide. However, synthesis of this was not great. To make TT was great, but to go to A or G, it was kind of very difficult, even if in the hand of people like Alice, who was experienced postdoc. And between Alice and Lillian, they tried to, instead of having this imide, to have maybe just to put 1 extra oxygen here or OCH2 more here, just to make the synthesis easy. The synthesis was easier, in fact, and we tried it. However, the biology wasn't as great as the imide. Then so our target now is to find one group which is to give efficiency similar to the imide, but better synthesis or easier synthesis. When I started my group in Southampton, I thought about this sulfate group, which I always thought about for ligation, in fact. When I started the ligation work, I started to think about the sulfate as a replacement of the phosphate. And this was suggestion from Barry Sharpless. And I always wanted to do it, but the problem was stability to oligosynthesis for short oligo, or the ligation wasn't great. Then I thought, if this is not really stable, maybe the LNA will make it more stable. However, I wanted to give it a go for the therapeutic. And it worked, but it is not that stable. But when you add the LNA, it make it more stable. With the help of the Bashir, who is here in the audience, and Pelma, I gave them some of these intermediates, and they followed on, and they made some oligonucleotide, and we tested them. The beauty thing now that this modification is easier to make than the imide because you can start with commercial available in the top, and this is few steps. However, this combination is not the best, but we need to optimize the condition. When we test them in biology, in fact, they are even better than the imide, which was hard to synthesize. So this is great news for us, which was just, the work is not published yet, but the manuscript is under review now. So we are very pleased with this addition, which is, as I said, working greater than the imide. And now in my lab in Southampton, EJ started to continue in this work because I want to go to disease model now. With the best one is this. If you have NH here, it will be easier synthesis. However, it is not available as LNA, which we think it is part of the binding affinity, which is higher by LNA. But we will explore this to see if one LNA here, to make the NH2 here, with LNA is a bit of few steps. So we want to test the biology. While we are doing the biology with this linkage as test, we want to test the NH as well. And the AJ in Southampton is doing this project. I want to explore, I always wanted to explore as well this, but we didn't have a chance, is to see the effect of this in therapeutic and see the difference between 1,4 and 1,5. I believe that the hydrophobicity of this triazole might have effect in the therapeutic. We preliminary tested it, but not fully tested. So I believe also the 1,5 might work better, but we will have to test it. This is Nina here, and some of my master students helped in the triazole project, but we don't have any biological data yet. With this triazole, I wanted to test it as well in messenger RNA. So how can we make RNA? We made the gene, but we didn't make the RNA yet. So we made short RNA, and we tested the translation, and it was very promising. So it is very high. However, to test it in cells, we needed it in a gene, in long enough gene. Making RNA is much, much, much harder than DNA, of course. So we struggled with a few postdoc has to take, and Cam, who was a PhD student. Anyway, I thought maybe we can stick with the short RNA using click, and I want to introduce some of the epigenetic modification, but then maybe we can put it in the middle of messenger RNA to have more impact. So we make long RNA with modification wherever we want to put it, but we will include it in messenger RNA. And when I started in Southampton, I said, okay, maybe I can start a new area, which is long messenger RNA modification, especially the 5' and the 3' end. With chemistry, you can introduce a lot at the 3' end and the 5' end. This project, Jolio, who is here as well, is helping in this project. This was a new area for me, and I wanted just in the start to see what is available for the 5' end and all that. Sebastian, who is here as well, he is PhD student in the Tom Brown group, and I used to co-supervise him through the Nature ETN project, which was including few of the academic in Europe, including Thomas Carrel as well. And Sebastian is very good student, but also he has experience in working with messenger RNA before in his previous lab. So I thought it is really great if he can be involved in this project to do some of this work, because of course, he knows a lot about this modification. So we started to make modification. We started to look into what is the 5' cap, what is available. And of course, the clean cap is the most efficient one. And he started to make it in the lab just because it was a little bit expensive. So he started to do it. Then while he's doing this, in fact, he has one intermediate, and he thought that, why can't we use this? It's just to introduce it with click modification. And then he started to think about having 5' modification here with messenger RNA, and then we need to see if it is affecting the stability, if it will be good enough to stabilize the messenger RNA. So this is the 5' cap. And while he's doing this, of course, you need this, you need the OH here. And we have chemistry in our lab, which how you change the 5' OH in solid support to azide. Then he thought, maybe we can just, so the the efficiency in transcription from DNA to RNA coming from, in fact, the priming here. So you have complementary here. So it gets rid of the kind of, when you start the transcription in the start, there is on and off of the polymerase. Then this will get rid of this heterogeneity. So he thought, if this is making this very efficient, why can't we just put this and start the transcription and see if this priming part will give this efficient transcription? It was a small idea from him, and he started to do this work, and then it been built up after that, idea after idea, and now he's got, in fact, very good project out of it. So he started to do this in solid phase chemistry, and of course we have synthesizer in the lab, and you can do this very easy in solid support. So you take this, you remove the DMT, you then add the second base, and then, of course, normally you just put iodine and then TCA to cleave the second DMT. And then you have this OH, and you iodinate it, and then sodium azide to form the azide here. And then you cleave this. So you don't need really to use fluoride here, although you have 1 TBDMS here, but you don't need the fluoride because you have here the 2' omicide. And because you have this group, you don't need really to worry about ammonia. So you can cleave it even overnight without worrying about ammonia to cleave the RNA, because you don't have here the OH, which can catalyze this phosphate. Sebastian, however, wanted to do it manually in like column, because he wanted it to be available for anyone in the lab to make it. He made this in large scale because he started with high loaded resin. And then he wanted to see really the idea, is priming helped? So we called our new primer Clean 3, and this is the clean cap, which is commercially available. And this is just the normal transcription with the triphosphate. And you can see here, of course, the messenger RNA is long, so you can't analyze it by mass. But what you can do, you can cut the first part using DNA enzyme, and then you have short one, which you can then analyze carefully to see the efficiency of transcription. So here you can see the result is very close to the most efficient cap here. So you can see the transcription is very efficient. However, the one which ended by the triphosphate, the normal transcript, you can see that the starting point has got a lot of truncated RNA in the start, which is well known in the field. So the idea worked, and the priming using this dimer worked really well. After that, we wanted to label it just to see the efficiency inside cells. So he labeled it with Alexa Fluor, and he used copper-free chemistry, which is very efficient, in fact. And you can see from the HPLC, this is the starting oligo. If you leave it 1 hour, you can see full conversion to the labeled one. And of course, again, you can cut it to analyze it carefully, and you can see that it is really efficient, the labeling really efficient. Right, okay, we needed after that to see this is the efficiency of priming. He used different kind of sequence to see the generality of this addition. And in fact, you can see it is very comparable to the clean cap. So we compare it to the clean cap, not to replace the clean cap, but we want to show that the efficiency is similar to the most efficient one in the literature or in the market. He wanted to show the stability in cells, so he used HIC cells, and he showed here that, in fact, the azide alone, so just the dimer alone, is not very stable. And you can understand this because it is very small. So the 5' end is just azide replacing the OH. So it is really small. So it will not protect the messenger RNA. However, when you leave it with bulky group, which in this case is the dye, you can increase the efficiency of the stability. So in fact, you increase the efficiency of the translation of this messenger RNA by increasing the stability of the messenger RNA itself. And we showed here that, in fact, the azide only didn't increase the stability, but when you have the dye, it increased the stability. And hence, we think that this is the reason why the translation is more efficient. As viability of the cells, he showed as well that it didn't affect, so the dye didn't affect the viability of the cells. Sebastian will talk tomorrow morning at 9 o'clock. If you want to know really more of the biology of this construct, he did a lot of work for the cap-independent translation. Because as I mentioned, we don't want to compete with the cap translation because it is very efficient now with the clean cap in the market. But what we want to introduce is the cap independent. So people, of course, study the cap independent a lot. However, we believe that it is not really that efficient study because you start with RNA, which at the 5' end there is the triphosphate. And with this triphosphate, it is vulnerable. So any study you are doing is not really that accurate because what you are doing, you are comparing different structure with different stability itself. So if you are comparing the cap independent with the cap independent, it is not that accurate because the cap dependent has got the 5' end protected. The cap independent, which happens to happen a lot in, not a lot, maybe 10% in human, the cap independent. And when biologists or the current available method to study this cap independent is not really that accurate. With our modification, we hope that this will help biologists to study the cap independent translation in more kind of accurate and efficient way. I tried to acknowledge the people while I'm talking, and now I need just to acknowledge the funding, and thank you for your attention.